This video is the answers to genetics problems 12 through 16, incomplete dominance and dihybrids with incomplete dominance. So, question number 12. In 4 o'clock plants, the gene for red flowers is incompletely dominant to the gene for white flowers. In the heterozygous condition, pink flower develops. When a pink flowered plant and a red flowered plant are crossed, we'd have big R, little r, and big R, big R. And if we do our Punnett square, we would have a 1-to-1 ratio of big R to big R, little r, and a 1-to-1 ratio of red to pink. The ratios for the phenotypes are always the same as the genotypic ratio in problems with incomplete dominance. Number 13, black feather color is incompletely dominant to white feather color in Andalusian chickens. Hybrids have blue feathers. Cross a blue and white chicken. So we have a blue chicken and a white chicken. And we would get big B, or big B, little b. Nope, scratch that. We would get big B, little b, or little b. So, there would be a 1-to-1 ratio of big B, little b, and little b. And there would be a 1-to-1 ratio of blue to white chicken. So you'd have a 50-50 chance of getting blue offspring or white offspring. This is the trickiest problem in this problem set. So in guinea pigs, coat color is incompletely dominant, and the heterozygous condition produces gray. And that is a blend of the black and white coat color. And the second trait we're going to be looking at, short hair, is due to a dominant gene, and long hair is due to its recessive gene. So a heterozygous short-haired male, so the genotype of the male, this is where it gets tricky, would be big B, big S, little s. So it's heterozygous for short hair, but has a black coat. Lots of students tend to put the heterozygous big B, little b, but that would give it a gray coat. And that's why we said careful here, because they see heterozygous short-haired black, and they think that the heterozygous applies to the black coat color. But the guinea pigs that are big B would have a black coat, big B, little b would have a gray coat, and little b would have a white coat. So the genotype of the dad is big B, big S, little s. Now we're going to cross that with a female who is heterozygous for short hair but has a white coat. So she would be little b because she has the white coat, and big B, little s because she is heterozygous for short hair. Now it's important to note that some students put the S's before the B's. It doesn't really matter which trait you used first, as long as you're consistent within your Punnett square. So if you did your S's before your B's, that's totally fine. So when we do this Punnett square, we would FOIL. We would have big B, big S, or big B, little s. One of the things that I always do when I have a problem like this, and there is a homozygous organism in the dihybrid cross, I always circle the alleles that are the same, and then I distribute like this. So in that case, you don't need a 16 square Punnett square. You're only going to need a 4 square Punnett square, and this can save you a lot of time if you realize that. So when you FOIL, you only have 2 options for each parent. You have, for the male, big B, big S, or big B, little s, and for the female, little b, big S, little b, little s. So when you fill in this Punnett square, you get a 1-to-2-to-1 genotypic ratio, but you get a 3-to-1 phenotypic ratio, because this individual would have gray short hair, this individual would have gray short hair, and this individual would have gray and short hair. This individual would have gray and long hair. None of the offspring would have black hair or white hair. They would all have gray hair, but then these 3 since short is dominant to long, would be short. Now, I want to note it for the record. If you did a 16 square Punnett square for this and reduced, it would be totally fine. It would just waste a lot of your time. As long as you always reduce your ratio, you're fine. Now, in bebops, red eye color is incompletely dominant to white eye color, and heterozygotes have pink eyes, and 2 antenna is incompletely dominant to the gene for no antenna, and heterozygous bebops have 1 antenna. By the way, this is not a real creature. These are problems that we've made up just for the sake of having more options for genetics problems. But the genotypes of both of the parents would be heterozygous. And when you fill this out, you FOIL and you have the same Punnett square as you would for a regular dihybrid Punnett square. And the only difference is that you're going to have a really ridiculous phenotypic ratio. So you would have 1 red-eyed with 2 antennas, 2 red-eyed with 1 antenna, 1 red-eyed with no antenna, 2 pink-eyed with 2 antenna, 4 pink-eyed with 1 antenna, 2 pink-eyed with no antenna, 1 white-eyed with 2 antenna, 2 white-eyed with 1 antenna, and 1 white with no antenna. If we were doing a quiz on this, we might give you a problem like this where you would have to set up your Punnett square like this, but instead of listing all of the phenotypes and ratios in this case, we might just ask you for all the possible phenotypes. We're not likely to ask you for a ratio like this. What we would be likely to ask you is to list all the phenotypes, and then we would pick one phenotype and ask what was the chance of getting that one phenotype. So for example, if we had done this problem on a quiz, you would get these phenotypes, but like we might ask on one of the problems, well, what are the chances of getting a pink-eyed bebop with 1 antenna? And then you would have to look at the Punnett square, find them, and make check marks in them and say, okay, there are 4 of them, 4 out of 16th is 1/4 chance. So for number 16, we have a codominant cross, and the only thing that's really different about the codominant crosses is the notation. You have an uppercase letter with superscripts. Now, if you cross a roan bull with a red cow, the roan bull is CRCW, and typically the R in these problems goes before the W if you're talking about the order, and we have a red cow. And so typically when you're doing these, the male's gametes go across the top and the female gametes go down the side. It just makes it easier for a teacher who's grading problems if everyone does it the same way. It's not something typically that a teacher would take off points for. If you had put the cow on the top and the bull down the side, you'd end up with the same answer. It's just sort of a standard way that it is done. So if we look at the possible combinations of gametes and zygotes, which are inside the Punnett square, we have a 1-to-1 ratio of CRCR to CRCW and a 1-to-1 ratio of red to roan.